Alright, so we saw up till here and we had made we have done the analysis using DSeq2, and the remaining part that we're going to do in this video is making a volcano plot to represent the upregulated and downregulated genes that are there in the result. So if you remember, we created this result where we used a p-value cutoff of 0.01, and if you pull up the summary here and you pull up the summary, you'll have this where you have the p-value which is set to 0.01. Previously it was 0.05, and you have these many upregulated genes and these many downregulated genes. You have these many outliers. Now we're going to take this and we're going to make a volcano plot out of this. So to do that, the first thing that I want you to do is install a package or a database, a human database package. So for that, you need to I'm going to copy paste a code snippet over here. So you need to get it from the bioconductor. This is the human database where we can annotate the genes based on their ensemble IDs. So this HS stands for human. If you're doing with mouse, it would be MM, so capital M and small m. So just check it out. What are the different annotations, keywords that you need to give for a particular organism? So once you install this is going to install two things: the database itself. It's going to pull out the database, and also it's going to pull out the annotation dependencies. So right now I'm going to comment this out because I've already installed it. Before you make the volcano plot, I want you to install this package. And now to go on with the volcano plot, first we'll annotate the ensemble IDs to the corresponding gene IDs or the gene names, and then we're going to put that particular column that we get from for the gene names into the volcano plot. Otherwise it's all going to be ensemble IDs all over the plot. So to do this, first we're going to call the library that we just installed. So these are the two libraries. The first one is the annotation DBI. The next one is the library itself. I think you do not need this because once you call in the library of the database, it automatically pulls in the library of annotation DBI. But just for the safe side, we're gonna say library for both of these and hit Command Enter. So now these are loaded. The next thing that we're going to do is take the result that we got and we're going to export it as a CSV file so that we can keep it and we can use it further to make the volcano plot. So what I'm going to say write.table, and inside this res 0.1. Actually this is going to be 0.01, but since I put this inside 0.1, I'm going to go with that. But the alpha value or the p-value is 0.01. Just keep that in mind. Just keep an eye out for that. So next we're going to say the file name. So I'm going to call it file equals to let's call it airway.csv because we're working with the airway dataset. So and there is this code. You have seen all this stuff in the previous videos, how to write a file into a CSV file. So code equals true, and for the separation I'm going to put a comma. So now it is ready to be exported. I'm going to hit Command Enter, and it is exported. I'm just going to show it to you. But before that, I'm gonna comment this. and I'm going to say exporting res 0.1 as a CSV file. It's very important to comment things out because you look at it after like a couple of years and you don't remember what exactly is the line of code doing. So it's better that you annotate like the main bits and parts of the code. Next, we're going to load the file that we just exported. So we put another hash and say loading the array file, and I'm going to say read.csv because this is a CSV file. Again, you have seen this in the previous videos. Array.csv, and what I'm going to do is I'm going to put this inside a variable called RNAseq so that it's easy to call it whatever we want. And if I do command enter, it's loaded. Just to view it, I'm going to say view RNAseq, and you can see that we have the result of res 0.1 as a table, and we have the p-value, the p-adjusted value, and the locked fold change. So we're going to work with these two, the p-value and the locked fold change. Alright, so now that out of the way, what we're going to do is, right now we have the ensemble IDs over here as the first column. What I want to do is I want to create a new column at the end where we have all the names of the gene or the symbols of the gene, because these ensemble IDs are very long and you don't want them to be showing up on your volcano plot when you do it. So to do that, I'm gonna say RNAseq, and I'm gonna pipe this into a function called mutate. And mutate is basically if you're creating, deleting, replacing a column, you can do that with mutate. So I think it comes from the dplyr package, which comes with the tidyverse. So I've already put in tidyverse, so that's why I can use it. So in type mutate, first I'm going to say the name of the column that I want to create. So symbol, if this column is not there, it is going to create a new column and put the values inside according to whatever we specify after the equal to sign. Or if the column is there already, it's going to overwrite it. Okay, so be careful about that. If the column is there, it's going to overwrite it and not create a new column. So inside symbol, I'm going to say map IDs. Inside this, I'm going to first put in the database that we just installed, db, and comma. Next is our key is going to be the row names of RNAseq. So this is the key, and I'm going to say there's another argument column which will be symbol, and our key type is going to be ensemble. So these are the different arguments that you need to give the database: the row names, where you're getting it, the column, and the key type, right? So we're mapping the ensemble IDs with the gene symbol. So this is the symbol, the gene symbol, and this is the ensemble ID. Now again, I'm going to pipe this and make a data frame out of it. Data.frame. Okay, now if I run this, it's going to give me a data frame, and to view it again, I'm going to do this. I'm going to hit view. Okay. So view. There's our new data frame, which has the symbol column, where you have the different gene symbols that we mapped out from the ensemble IDs. So that's what this column and key type means. Now, since we're ready with that, we can go and directly start making the volcano plot. So before you make the volcano plot, you need to install a package that will give you, that will help you make the volcano plot. It's called Enhanced Volcano. So all you need to do is go and again say install packages, Enhanced Volcano, Enhanced Volcano. Okay, it should be in quotes. I'm not going to do that. This should be in quotes. So one quote up here, one quote up here, and you can install the package. After that, you're going to call this package, saying library enhanced volcano, and it's going to be loaded. Now you can start making the volcano plot. It should be enhanced volcano. This is the function that we're going to use. Alright, so what I want to do before starting this, I'm going to put this whole thing inside, and I'm going to reassign this to RNAseq. So I'm going to delete this view. Command enter. Now I have everything, whatever I created, along with the gene ID, the gene symbols inside RNAseq again. So for enhanced volcano, the first argument that we need is the data frame RNAseq. The next thing that we need is the labels, and this is a little bit tricky. I tried a lot of things, then I came up with this. You cannot directly put symbol. Okay, you cannot do this. It's going to throw you an error. What you need to do is you need to subset the column from the original data frame. I don't know why, but it is like this. So it's like RNAseq, you subset it with symbol. Okay. So this is how you need to write it. Your x-axis is your log2 fold change. Careful with the spellings. Your y-axis is going to be p-value, and up to this we're good to go. We can just look at the volcano plot. Alright, so you have a nice looking volcano plot. So you have the gene symbols over here. If you wouldn't have changed it, it would have been all ensemble IDs. Long ensemble IDs on the plot does not look good. So this is the default cutoff, where it is 0.5. The upregulator is 0.5, the downregulator is minus 0.5, and you have a p-value cutoff of 0.1, I guess, because we had set it to 0.1. You can change the p-value, the horizontal line, by saying p cutoff is equal to, you can specify whatever you want, 0.01. and f cutoff is 0.5. You can increase that, decrease that according to your needs. And another thing that you wanted to do is point size. So the point size, it's going to be, let's make it 3, and the label size, let's make it 5. Now if we put it, it's going to be a little bit smaller, or we can make it 7, just view it. Yeah, it's bigger now. Next, if we want to change the legends over here, so I think it's going to be legend labeled, and we're going to pass in the values in a concatenation. So the first one would be this, then this, then this, then this. So the first one remains as non-significant, or we can say p less than 0.01 and fc less than 0.5. So there's our first one. The second one, the green portion, it's p less than 0.01, only that. Next, there's p greater than 0.01. This is also p greater than 0.01. The blue part is p less than 0.05, so 0.01, and fc fold change is less than 0.5. Okay, so, or we can say, yeah, let it be like that. And finally, the last one, it's differentially expressed. So now if we run this, then you see all this stuff over here, and you can change this according to what you want. And this is just an example. You can check out the enhanced volcano documentation on the website, and that's going to give you a lot more arguments that you can put. Here you can change the shape of the symbols, and you can do a lot of stuff. You can change the title, the subtitle. Yeah, so that's about it. This is how you can make a volcano plot out of your RNAseq data, and you can get it publication ready. Alright, I'm going to see you in the next one.